Yeah, good evening students. Now I'm going to say about the sequence elements used in bioinformatics. So why are you using these sequence elements? The sequence elements can be used for the to identify your research results. So here, so have a sequence. Suppose you got a sequence, you identified, you got a tissue. From this tissue, you isolate the protein. Those protein sequence has to be identified. So those protein sequence means enzyme sequence has to be identified. So for this case, we are going to use the software tools. That's called search engines. We are going to use it. So bioinformatic tools we have it. Those are called as pairwise sequence element and multiple sequence element. So pairwise sequence element means so for this pairwise PSA and MSA multiple sequence element. Both we can use for the protein sequences and DNA sequence. We can use it. So now we will go for the pairwise sequence alignment. So what here we are going to do that one. So we are going to take a sequence. We are going to take a sequence. Those sequence you are going to compare with already existing sequence. So suppose you have got a similarity. Suppose glycine is there. For example, protein sequence. Glycine is there. You got a glycine in first position, and second position you got a phenylalanine. Same you got it. Like this you got the same sequence from your results. So we can got the 100% accurate results. We can say that 100% similarity. Then we can claim that my sequence is similar to that one. So finally we can conclude that so this sequence will be similar to that one. So I can say that my result is good. So finally we can say that so these results, based on the results, I can submit my sequence in my NCBI databases. So here in NCBI I said you submission we can submit your own sequences. Those sequences can be deposited with your name. So here the pairwise sequence element, we can search your sequences. Suppose the normal sequences we got a similarities, 99% similarities. We need to give your results predictions. Then after the result giving the predictions, then we can go to the submission. Those are the use of our pairwise sequence element. So pairwise means we are going to compare only two sequences. One is a query sequence, already exists a sequence. So our query sequence, our sample sequence is called a query sequence. Then already existing sequence is called a standard sequence. You can say that. This is called as pairwise sequence element. So here, this pairwise sequence element can be done with different softwares. We can use it. We can use the what is that? We can use the dot matrix. We can use it. Different types of softwares we have it. I can say that's one latter. So now again I'm going to multiple sequence element. What is my multiple sequence element? Suppose I have more than two sequences. So I want to compare that sequences. So to compare that sequences, I'll have to be align that sequences. The alignment means I'm going to arrange that one. So best example I can say that. So here first sequence I have it. Here we have different methods we have it. One is I can see this one here. So one is sum of the pairwise method. Next one is a progressive alignment method. Then iterative method. Then we have a profile element method. Multi-dimensional programming we have it. Multi-dimensional programming. Then next one, the first we'll go for the sum of the pairwise method. Sum of the pairwise method means we're going to add the sequences. We're going to give the score to the so two sequences. Those score, based on the score, we are going to put the ranking. Clear? So suppose I have a sequence. Now first your standard sequence I have it. For this I have given other sequence, my query sequence. I got the 100% similarity. So my first sequence I can put in the first number one because which one is got a more score, like our student score. Suppose a student they got secured more marks in their exams. We are going to give the first rank. Then less marks compared to the first one, you can give the second rank. Like this, you are going to give the ranking. So here also sum of the pairwise method. So you're basically this sum of the pairwise method means you're going to compare the methods, can give the compared sequences, then you'll give the based on that one we can give the ranking. Next one is what is that we have a progressive alignment method. Progressive alignment means so here we're going to take the two sequences. You're going to take the two sequences that can be standardized. So two similar sequences you're going to take it. That can be standardized. Then I'll go to after standardization of these first two sequences, I'm going to third sequence. So then I can go for fourth sequence. So here this standardization has to done it. So that's called as progressive. Means one by one. Means progress means you're going to increase this. You're going to increase your sequence pattern. This is called as progressive alignment method. Next one is iterative method. Iterative method means what I say that repetition. Repetition. Suppose we got a less sequence, we got less score, we got it. Then again we will go for the repetition of that sequences. So we can add we can add some of the sequences, we can delete that one. So our main motto, our main aim in your alignment is to increase your score. Suppose all the sequences are almost all, we can say that. So differences we can find it, but of the some of the sequences, suppose we got a liver, I have a liver. From the liver I got a isolated protein, liver protein from the one human being. So those sequence, liver protein sequence, will be similar to other human beings protein sequence, liver protein sequence. So we don't find much differences. So here, we can get based on the results, we can have the deletions, and maybe deletions we can do that. So we can increase the score. But why you are going to have the sequence comparison? So we can identify it may be any evolution modifications in our sequences. We can identify the evolution modifications in our sequences. So here, like this, we can do that sequence alignment. That's called iterative. Means you're going to repeat that sequences. Then next one is profile alignment method. Profile means you're going to consider the sum of the sequences. Means 10, 20, 30. Means first one is 10, second 10 means 1 to 10, 10 to 20. So like this, you're going to get a profile. One file you're going to consider. This one you're going to consider as a one file. So in this file, we're going to compare the sequences. Then we can give the score. Means first sequence and second sequence. When the first 10, so you are going to give the score. Then you again the second 10 to 20, first sequence and second sequence, you got a score. So like this, you're going to put it in a profile element. This is called as profile element or profile score calculation. Then process calculation. Process means what is about I wrote the sequence. So first position of my sequence, what is that one? In second sequence, what is the first position of my sequence? We're going to compare this first position, means each and every site. So your sequence maybe have a 100 base pair length. In this 100 base pair length, so first, what is the first one? For my case, what is the first one? Mine. So already standard we have it. In this standard, so first sequence is first base pair means what is that? Suppose I can say that nucleic acid A, I have a A. But in my case, my sequence, my core sequence, I have maybe T. So I'll say that okay, this process is different. So this process is different, so I'm going to change that process. So when you change one process, I can get the new sequence pattern also. I can get the new sequence pattern. Like this, you're going to do that your sequence alignment. The next one is multi-dimensional programming. Multi-dimensional means simply we are going to put the whole sequences. We're going to identify the multi-dimensional, multi-way